Welcome to the new semester. We are going to start the first chapter of the semester, enzymology. Enzyme may be the most important term a biochemist has ever used. Enzymes are very important biological catalysts present in all living organisms. And clinically, industrially, pharmaceutically, or in all aspects of life, they are very much important for us. Actually, we should know that history of biochemistry is really the history of enzymology itself. So, what's it actually? All living cells contain enzymes. Remember, all living cells contain enzymes. Remember, cells are making up tissues and tissues are making up organs. And at the basis of the functional aspects of a living thing, there are the enzymes. And they are extensively used in laboratory works also. Enzymes are biological catalysts, or we should say they increase the rate of biochemical reactions under ideal environmental conditions. What's an ideal environmental condition? An environmental condition most suitable for the functioning of enzymes. For example, you know that our body temperature normally is 37 degrees Celsius. And most of the enzymes of our body are functional to the maximum extent at this temperature. Okay? And there are some other conditions also which affect the activity of an enzyme. For example, pH is there. And also, the concentration of a substrate available is there. And in most cases, presence or absence of modulators is a very important parameter. And again, presence of inhibitors. So all these are very important parameters to decide the ambient environmental conditions for the activity of an enzyme. And what are enzymes? Biochemically, most of the enzymes are proteins. Look here, biochemically, most of the enzymes are proteins, but don't say all the enzymes, because at least a few enzymes are there which are not proteinaceous in nature. So, at very high temperature, these enzymes may get denatured. Denatured means they get destroyed, and in most cases, irreversibly. And at low temperature, they lose their activity. Also, this is what we are doing in our home, in our refrigerators. We are used to preserve food material in refrigerators because in the low temperature of the refrigerator, the microbes which are responsible for the spoilage of the food are not able to function properly. And cooking, etc., makes meat tender because of denaturing and softening of that proteins in it. And what are the various factors that affect the activity of the enzyme? See, I told you just earlier, temperature is there. Too high temperature or too little temperature won't help a proper functioning of the enzyme. And also pH is there, and concentration of substrate is there. And enzyme, I told you enzymes are proteins, so enzymes are having a three-dimensional structure. A complex three-dimensional native structure is there for every enzyme, and using modern laboratory techniques we can find out the structure, and if there is any alteration in this structure, that enzyme will not be functional. And how is an enzyme functioning? Look here, what you see here is actually a graph. You have seen this graph earlier. On the vertical axis we have energy. while on the horizontal axis we have the course of the reaction. You see, here is the ground state, which means I want to tell you that this is the energy of the substrate before the reaction, and this is the energy barrier to overcome. What does it mean? Only those substrate molecules which have this much energy, this is the energy difference, this much energy can take part in the chemical reaction. I told you this is the energy of most of the substrate, but those substrates will be able to take part in the chemical reaction only if they have this much energy. So this energy difference is actually the activation energy. This is the activation energy. So what is activation energy? Activation energy is the difference between the actual energy of the substrate and the energy to possess, I mean the energy required to take part in the chemical reaction. So this uphill is actually called activation energy. This energy difference is called activation energy. Only those substrate molecules which have the activation energy or which attain the activation energy will be able to take part in the chemical reaction. And how is an enzyme increasing the rate of the reaction? You know, very simple. The enzyme is lowering this activation energy. Say this is the amount of, this is the activation energy in the absence of the enzyme, but when an enzyme is present in the medium, it is lowering the activation energy just like this. The enzyme is tremendously lowering the activation energy and thereby enabling most of the substrate molecules to take part in the chemical reaction. And there is a term called substrate. What's a substrate? A substrate is the molecule upon which the enzyme is acting. In chemistry, you call it reactant. So substrates are the molecules upon which the enzyme is acting, and there is a site upon the enzyme where the substrate is binding. That site is called active site. What is active site? An active site is a pocket or a crevice on the surface of the enzyme where the molecules are binding, attaching. You see here, this is the substrate molecule. This substrate molecule is able to bind here. So the active site of the enzyme is complementary to the shape of the real substrate. This is the mechanism of action. And there should be a perfect fit. What does it mean, perfect fit? The substrate should fit perfectly into the active site of the enzyme. Only then the reaction will take place. If there is an imperfect fit, or if the substrate is not matching with the active site of the enzyme, the enzyme reaction will not take place. The substrate is not converted into product. Actually, there is a concept called induced fit. We will discuss induced fit hypothesis later when we come to the mechanism of action. And sometimes, you know, the activity also requires something called cofactors. The activity requires cofactors. What's a cofactor? A cofactor is another molecule, a non-proteinaceous molecule. Sometimes an organic molecules, sometimes inorganic ion, for example manganese, magnesium, etc., required for the activity of the enzyme. So the proteinaceous molecule without the cofactor is called apoenzyme. The term is not given in this slide. Apoenzyme is the proteinaceous component of the enzyme in the absence of cofactor. So when the cofactor is binding with the proteinaceous factor or proteinaceous fact, that the component, we call it holoenzyme. Holoenzyme is the functional form of the enzyme. And you can see here a term called coenzyme. All coenzymes are cofactors. Actually, coenzymes are organic cofactors. Coenzymes are organic cofactors, and they are required for the activity of enzymes. You might have heard of coenzyme A, etc. That's a good example of a coenzyme which you have studied in your lower classes. And when cofactor is there, the holoenzyme is able to function properly. And sometimes the activity of the enzymes may be inhibited. Such molecules which are causing the inhibition of an enzyme activity is called inhibitor. Okay, and inhibitors are of different types. Sometimes there is a competitive inhibition. What is meant by a competitive inhibition? This inhibitor is structurally similar to the substrate. So naturally, when the substrate and the inhibitor are similar, the enzyme is in confusion. So there is going to be a competition between the substrate and the inhibitor for the active site of the enzyme. Here you can see this inhibitor, this green colored inhibitor is really matching some part of the active site of the enzyme. So if the inhibitor is binding, the reaction will not take place. On the other hand, if the substrate is binding, the reaction will take place. Okay? And how is it functioning, or what are the functions? Sometimes synthesis of biomolecules from the raw materials. Synthesis of biomolecules from the raw materials. For example, protein synthesis, synthesis of starch, synthesis of complex lipids, etc. from their raw materials. Synthesis of DNA. You might have heard of DNA polymerase enzyme. That enzyme, DNA polymerase enzyme, which is responsible for the synthesis of DNA from deoxyribonucleotides. So good example for an enzyme which is a synthetic enzyme. And breakdown of biomolecules. Complex biomolecules are broken down into simpler forms. For example, digestive enzymes. These are the functions of enzymes. All enzymes function either by synthesizing larger molecules or by breaking down larger molecules. And also, sometimes enzymes are transferring also. They are transferring some molecules or some part of molecules, etc. We are going to study these functions in the classification of enzymes in detail. And what are the applications of enzymes? In biocatalysis, in biotechnology industry, in diagnostic pharmaceutical industries, in food and beverage industries, in animal feed, food, etc., and in pharmaceuticals, in biofuel synthesis, household materials. In almost all aspects of human activity, now we are using enzymes. Do you know that detergent powder which you use sometimes to wash your clothes contain enzymes? They're unbelievably, these enzymes are actually penetrated into all aspects of human civilization, that we cannot live without them. Do you know that curdling of milk is an enzymatic activity? Do you know that we convert food materials into pickles in order to prevent the enzymatic activity of that degrading microbes? So like that, enzymes are actually used in all aspects of human activity. Sometimes we promote the activity of the enzymes, sometimes we want to prevent the activity of enzymes. For example, in pickling of food, in refrigeration of food, etc., we are trying to prevent the activity of the enzymes. And disassembly of complex plant cell wall in biofuel production. When we are converting cellulose, etc., in the plant materials into ethanol, we are using enzymes. This is just one of the examples. In disease diagnosis, you might have heard of ELISA test, etc. You might have heard of RT-PCR test, etc., which are used in a COVID-19 test to make specific cuts in DNA molecules. You might have heard of restriction endonucleases. We are using enzymes. In recombinant DNA technology experiments, we are using enzymes. And amylase is a common enzyme present in your saliva. Lipase is an enzyme present in your alimentary canal. Actually, it is produced by your pancreas. Proteases are digestive enzymes present in your alimentary canal. You might have heard of, various protein digesting enzymes in our body. For example, pepsin is there, and trypsin is there, and chymotrypsin is there, etc. And different proteases are there.